the Darwin Journal. All right, so let's keep this simple. I'm going to have the topic and what kind of topic it is in that corner, and the artifact in this corner. So let's get started with Unit 1. So with Unit 1, all my artifacts are actually TRGs, so for my justification, I wrote them all, and they all basically explain every single topic, so I don't really think I need to justify most of them for Unit 1. So let's start with topic 1.1, which is the structure of water and H bonding. And the main topic of this topic is water. So with this topic, it's mainly about just H bonding and how it connects like water molecules. Like it connects two same water molecules with either a cohesion or adhesion bond. A cohesion bond has, it connects like water and water, and an adhesion bond connects water and something else. Most of the time you're gonna find a cohesion bond, and with cohesion, you'll find it like a lot of properties with water. It creates like surface tension and just a bunch of other properties with water. Moving on to topic 1.2. Topic 1.2 is basically all about the elements of life. Nothing too complicated here. Basically saying we need like energy to move around, do our daily stuff and stuff. But it actually starts to slowly introduce us to the whole focus of the unit, which is macromolecules, carbohydrates, lipids, proteins, and nucleic acids, stuff like that. It also starts to slowly introduce that into the wider picture of the unit. Quick little side note, I forgot to add, topic 1.2 also goes over how like carbon molecules like connect stuff in like different branches and patterns and like circular objects. Yeah, it also goes over that. Forgot to mention it. Moving on. So topic 1.3, this is where we truly start getting into the main point of the topic, which is macromolecules and how they work and how they have monomers and polymers. Monomers being the stuff that makes up the polymers. Polymers are just a big chain of monomers. And you may notice I have my notebook here because I took one too many takes trying to say the monomers, and I could not remember for the life of me how to say them, so I'm just going to read them off the notebook. So for the first polymer, we got carbohydrates, and the monomer for carbohydrates is monosaccharide. For the next polymer, we have proteins, which the monomer is amino acids. The next polymer is nucleic acid, and the monomer for it is nucleotide. And the next polymer is lipids, and the monomer for it is fatty acids. And we also go over dehydration synthesis in this topic, basically saying the removal of water creates the bond between these monomers that connect them into polymers. Topic 1.4 is all about carbohydrates, and out of the four macromolecules, they probably have some of the most stuff to do because they make up stuff like starch, cellulose, and glucose, main functional components of the body. And these are all made up from carbohydrates, all because of how they are structured as polymers. They could be like square shaped, they could be branches off, or they could be even circles like starches. And it basically just goes over how the shape of a polymer can change what the actual macromolecule does and all the functions of carbohydrates in the body. So I'm just going to combine topic 1.5 and 1.6 into one big section because that's how they were in our class. And yeah, this is basically the structure and function of the biological materials in the body. It's basically just going more in depth into each macromolecule itself. And we're going to start with lipids. Remember what I said earlier, that lipid about monomers and polymers being like just one in one, that monomers are just a big chain of polymers? Well, lipids don't follow this rule. Lipids are just on their own, and they're just singular components by themselves. So they're not really any monomers for lipids, they're just polymers. And their main function is to store energy for like the body. They just store it up, and that's where they keep it. Next up, we have nucleic acids, which is basically the DNA and RNA of the body. They store and transmit genetic information of the body, and yeah, they're just what store what is you in the body. Proteins, their function in the body is to basically just keep the structure and control the body and make sure everything's going on as it should in the body. Carbohydrates is basically the energy source. It's the power cell. They keep the body going. They give the energy to everything else. They produce stuff like glucose and starch, which gives energy to the whole body. And that about wraps up Unit 1. So let's go into Unit 2.
All right, so I'm in the process of editing this video, but I kind of forgot to include topic 1.7. So I'm just going to add it in right here. Topic 1.7 is all about proteins and how they work. and stuff. So with proteins, you got their building blocks, which is amino acids, is what the protein's made of. And these amino acids change everything about the protein, but they're all based around one central carbon or whatever. And all this stuff is connected by peptide bonds. But whatever's connected to this carbon changes what the protein does. Stuff connected could be like polar, non-polar, or charged stuff. And the way how these charged polar and non-polar molecules work together is by like they're changing their shape, size, can affect anything that the protein does. And this protein basically is like the control thing of the body and makes sure everything works out how it does. Just a quick little side note, the artifact for this topic is a bell ringer we did about the structure of proteins. And it makes sense because I just did it myself. All right, that's about it. Moving on to Unit 2.
All right, so this unit's going to be like the last one. Most of my artifacts are just going to be TRGs, but I also do have a lab and a few topic questions sprinkled in. So let's get started. This unit's mostly about organelles and stuff and how they work in harmony together just to keep the body going. And first, in this unit, we're going to have to learn the organelles, and that's where we get into topic 2.1, the structure and function of these molecules. Here we learn stuff about like the Golgi complex, the lysosomes, the ribosomes, all that kind of stuff, and how these organelles work together and all the specific functions they have in the body, like they transport molecules, proteins, all that kind of stuff in the body. Keep it all running. Topic 2.1, this is all about the cell size of these organelles. So these organelles, they all have their function, but they also have to have time to perform these functions. And the size of these molecules or organelles have a big factor in it. The bigger the organelle, the less efficient it's going to get. It's going to get slower. Sure, it could take in more stuff, but it's going to take longer for that stuff to come out. But the smaller the organelle, the faster the stuff's going to get turned out. So you're going to have a lot of small organelles rather than just 3 like big organelles. So smaller is better. It's all about that and like how the size and the shape affect how these organelles function and provide to the body. So for topic 2.3, I would usually have my artifact up there, but it's currently not there because it's locked. I could not get the topic questions for topic 2.3 and 2.4, so I'm just going to talk about it. Topic 2.3 is about the plasma membrane, like the membrane that's like in the middle of the cell that like keeps the barrier or whatever. It's made of like phospholipids. It has a hydrophilic head, meaning it's like likes water, attracts it, and hydrophobic tails, which they don't like water at all. The hydrophilic head is going towards the outside where all the water is, and it's keeping the hydrophilic tails away from all the water. But this plasma membrane isn't just made of phospholipids. It also has proteins scattered all throughout it, and these proteins could be hydrophobic, hydrophilic, or a bit of both actually. But these proteins, they basically do everything in the plasma membrane. They like transport stuff and keep the structure of it all sound. And that's basically what topic 2.3 is about. Topic 2.4, once again, no article on the top right. And this is all about membrane permeability, which is basically what can go through the plasma membrane. And it's basically like carbon can go through it, oxygen can go through it, small stuff like that. But bigger stuff like ions in molecules, they need to go through transport proteins to be able to enter the cell. But some molecules have more than just the plasma membrane just blocking stuff in. They also have cell walls that can block a number of stuff, like an excess amount of water coming into the cell, stuff like that. Topic 2.5, we got the artifact back now. This is all about membrane transport and how these things pass through the plasma membrane. And they happen basically out of 2 different things, either passive transport or active transport. With passive transport, it just happens on its own. It's a built-in feature of the plasma membrane. Molecules will go in and out of it, just no problem, nothing required. Active transport, that's where you're actually going to start needing stuff. That usually is through like transport proteins. These require ATP, which is just a fancy way of saying energy. These need ATP to go through the plasma membrane. They need it. They can't go through it without it. And there's 2 final things for things how it could get through the plasma membrane. That's going to be endocytosis and exocytosis. With endocytosis, it's going to be taking in materials into the plasma membrane, like large amounts of materials. And with exocytosis, it's going to be releasing large amounts of materials from the plasma membrane. Topic 2.6, facilitated diffusion. This is basically all about what is being moved inside and outside of the plasma membrane. the goal of facilitated diffusion is to remove stuff from high concentration areas to low concentration areas. And these can only be done through transport proteins. You need a transport protein to do these. And these can basically just moving things from high concentration to low concentration and like balancing stuff out, like make stuff even. Ions can also move through transport proteins. They can only move through transport proteins. But if you move too many ions at once, you get polarization in the transport protein, making it unfunctional for a while. Topic 2.7, osmosis and tonicity. This is basically all just about the movement of water. And for my artifact, it's going to be a lab we did about carrots and soaking them in water and seeing how the water changed the carrot and the water around it. So osmosis is basically just the movement of water to like places that need it more. So it's going to be moving it from high concentration areas of water to lower concentration areas of water. And this is just making sure that in the cells don't get too much water and some cells don't get more water than they need. It's basically perfect amount, not too much, not too little. Topic 2.8, the mechanisms of transport. This is all about transport proteins, specifically for active transport, because passive transport, they don't really need any of this. But with active transport, it requires energy, which is an important part. So for these active transport things, they want to do a method where they'll get more energy later on so they could keep doing their active transport. And for active transport, it's more for larger molecules that just can't do it passively, like just go through the cell membrane. They need like a transport protein to be able to do it. Topic 2.9, cell compartmentalization. This is basically just how fast the cell can make all these reactions happen. And this is all with surface area. This is why you'll see in like the mitochondria, there was a bunch of folds in the cell. The more folds it has, meaning more surface area it is. The more surface area you got, the more room for reactions there. This keeps everything all organized and stuff. One reaction happens at a time in its own separate place. Everything has its own spot to work with, and everything is organized. Basically, more surface area, more reaction space, and cleaner reactions at a faster rate. Topic 2.10, the origin of cell compartmentalization. So the two main things we want to look at here are prokaryotic cells and eukaryotic cells. Prokaryotic cells are their own thing. They have their own kind of structure and stuff. But eukaryotic cells, they already have a bunch of internal organelles and membranes and stuff that all work together. But what happens is that eukaryotic cells will eventually consume prokaryotic cells to create more forms of just stuff in the eukaryotic cells. And what the prokaryotic cells become in the eukaryotic cells is stuff like chloroplast and membranes, which helps the eukaryotic cell thrive in the system. And that's about it for Unit 2. Moving on to Unit 3. Unit 3. Most of my artifacts for this unit are going to be topic questions that I answered myself and reviewed, and one of them is going to be a TRG, which is for topic 3.1. I actually have a TRG for it right there. So topic 3.1, this is all about enzymes and how they affect the body. So normally on your own, with your body doing the reactions it would, you would die at the pace that those reactions goes. Your body can't sustain itself. Enzymes, though, greatly increase the speed at which these reactions happen. They greatly reduce the activation, like the energy it costs, like to activate this reaction. But enzymes do have a flaw, though. They can only work with specific substrates. Substrates are what goes into the enzymes to produce energy. And each enzyme has their own specific, like, docking area that a substrate can go into. And only one for one. You can only do one substrate for one enzyme. And yeah, that's about it. Topic 3.2, the environmental impact of enzymes. So remember how I was just talking about how great enzymes are? Yeah, they also have a lot of flaws. They're quite picky and they're pretty fragile. What I mean by fragile, if you change basically anything about them, their efficiency rates go drastically down, and sometimes they just don't even work. Stuff from like the temperature of the area to the shape, or just kind of what kind of molecules are around it. Specific example, the enzymes in your stomach, they like your stomach acid, so they like to stay there. Stuff like that. Also, there's also stuff called inhibitors that could also block enzyme functions. There's competitive inhibitors that could literally just clog up like the docking area of the enzyme, which making it useless. And then there's a non-competitive one that literally just binds itself to wherever it wants on the enzyme, completely changing its shape and stopping its function. So bottom line, enzymes are pretty fragile and they're kind of picky on what they take as well. Topic 3.3, cellular energy. This is basically what all the energy that these enzymes make, this is basically what they do. Everything in the body is either going to do one of two things. It's either going to take energy in and use it, or it's going to produce this energy. And if this flow stops, you're just going to die. You need energy, and you need a constant flow, consistent flow of it too. And this is where these cellular energy comes in. Everything in the body needs to have an order. So it's like basically a factory line. One thing takes in something to make another, and then whatever that product is, it goes into another process to make a new product. Just constant refueling the cycle. Basically, everything has a purpose to fuel something else, and that fuel is used for something else until it finally gets back to them where it started. Topic 3.4, photosynthesis. This is probably a topic we've learned about 100 times. Light energy goes into a plant along with water, CO2, and you get oxygen and sugar comes out of it. It's pretty simple. So the light energy, it goes through the chloroplast, and this chloroplast turns it into ATP that the plant could actually use. And this goes back to what we just talked about in unit 3.3, in this like continuous cycle. This ATP goes in with the CO2, goes out, creates a product, and enough stuff to start the cycle all over again, along with the new stuff coming in with the CO2 and sunlight, just to create the sugar and the oxygen coming out of the plant. Topic 3.5, cellular respiration. We did it. We're at the last topic. We're almost done. All right, topic 3.5. This is all about cellular respiration. This is basically kind of wraps up everything we learned in Unit 3. This takes in like recycling energy, the energy you get from enzymes and photosynthesis. It all kind of brings it all together in a cycle. Everything on the earth does it. I do it, plants do it, and you do it as well. This is basically just the cycle of energy, photosynthesis happening in plants. This is all that combined into one big cycle of reusing energy and taking in new energy. And we also have electron transport chains, which takes the energy that's produced to transport electrons throughout the body to help the body function, produce new stuff. And that concludes the Darwin Journal. That was fun. It's currently midnight as I'm filming this. It's gonna be fun editing this video. That's about it.